Today I'm going to talk to you a bit about a mechanism of regulation of gene expression. Obviously all genes and their products, they need to be regulated in terms of how frequently they are transcribed and translated. This is done in many complex ways. I'm going to look at one of the simplest one, which we find in prokaryotes. This is the lactose or Lac Operon. So the Lac Operon is very well studied in E. coli, one of the model organisms. An operon itself consists of the promoter and operator regions. These are the ones that regulate the transcription of the genes. And then these three genes, LacZ, LacY, LacA, are all called structural genes. So an operon consists of regulatory genes and structural genes. Structural genes being those that when they are transcribed and translated form polypeptides. There's also another regulatory gene here. Now I've put this regulatory gene separated out from the rest of the operon. Now if you were to look at the genome of E. coli, it's basically a circular ring of DNA, and you could have these operons. Let me put in the promoter, the operator, and I'm going to go and do, here's LacZ, LacY, and finally LacA. Now, in this circular DNA, when transcription occurs, it will go round and round and round the RNA polymerase. So if the regulatory gene is here, when that gene is transcribed and the product is formed, that's completely independent of what is happening in the rest of this operon. So the regulatory gene product is transcribed and translated independently of any of these products. Let's look at all of the components of the Lac Operon and their functions in the system. So the first component that we have of the Lac Operon is this regulatory gene. The regulatory gene, when it is transcribed and translated, forms a repressor molecule. And this repressor molecule will be transcribed and translated completely independently of all of these structural genes. The repressor molecule will be present in the cytoplasm of the bacterium constantly and is able to interact with the other regions of the operon. The promoter region contains the site where the enzyme RNA polymerase should bind. So it will have a complementary shape to the active site of RNA polymerase, so that we can draw the enzyme around this section of the DNA strand. The next section here is the operator. The operator is a section of DNA that has a region complementary to the shape of the repressor molecule, so that when the repressor molecule is present, it binds to the operator region, making it impossible for RNA polymerase to advance beyond the operator region of the DNA. So it puts a physical block between RNA polymerase and these structural genes. So if the repressor molecule is present, no transcription or translation of the Lac genes will take place. But what is the point of having all of these genes if they're never going to be transcribed? And this is where the clever aspect of the regulation comes in, because let us look at what happens if lactose is present in the growth medium around E. coli. So lactose, you should remember from studying biological molecules, is made up of one molecule of alpha glucose joined to one molecule of galactose with a glycosidic bond formed by condensation. Lactose is not a very common source for E. coli, so they do not need to be able to produce the enzymes that would allow them to digest lactose unless it's present. And this is where the clever feedback system comes in. If we have molecules of lactose in the cytoplasm, because they will be able to enter down their diffusion gradient through lactose transporters. So here's all the lactose. Okay. What you may notice about the lactose here is, especially since I've drawn it quite deliberately like this, there is a spot on the repressor molecule where lactose can bind. And if lactose binds to that repressor, it changes the shape of it. You see, it's become a bit stretched out of place, so that it now no longer has the complementary shape to fit in the operator region of the operon. So if there's lactose present, these repressor molecules will undergo a conformational change when they bind with lactose, and they will not be able to bind to the operator anymore. What this means now is that RNA polymerase has no impedance towards crossing through this operator region and then transcribing and translating LAC-Z, LAC-Y, LAC-A. These are the three structural genes that give rise to proteins that help to break down lactose, that help to increase the rate of lactose transport into the cell. And so when these genes are transcribed, lactose can be metabolized. If we look at some of the functions of these structural genes here, we have beta-galactosidase. Beta-galactosidase will break down the lactose, it will hydrolyze it into glucose and galactose. Lactose permease, as you might expect because of the name permease, actually forms more channels for lactose to be transported into the cytoplasm of the bacterium. So we're going to increase the rate of diffusion of lactose into the cell and increase the rate at which it can be broken down and metabolized. So this is a direct response by the bacterium. Increased concentrations of lactose in its environment means an increase in the concentration of lactose in the cytoplasm through diffusion, which results in the blocking of this repressor molecule so that RNA polymerase can transcribe the genes that will form the enzymes that will help with the metabolism of lactose. If we then go back and look at what happens when all of this beta-galactosidase and lactose permease has ensured that as much lactose as possible enters the cell, it's being broken down, so the concentration of lactose is going to decrease as these structural genes and their products are acting on the lactose molecules. And so what happens as these disappear, as have been broken down into galactose and glucose, is that we have repressor molecules now being produced that have not had their shapes distorted. So because now the concentration of lactose is low, the repressor molecule can bind to the operator region and prevent RNA polymerase from transcribing the structural genes. So this is basically an energy saving mechanism. There is absolutely no point transcribing beta-galactosidase, the metabolic energy with the ATP involved in that set of reactions, if there is nothing for it to act on as a substrate. So it's a very simple negative feedback here. It works brilliantly with lactose and with the E. coli. In summary, Operons consist of regulatory genes plus structural genes. Structural genes, when transcribed and translated, form enzymes or proteins or other polypeptides that may need to undergo quaternary structural changes. A repressor molecule is produced, which binds to the operator region. RNA polymerase binds to the promoter region, and if the repressor is present at the operator, cannot transcribe these genes. If lactose is present, the repressor molecule shape is altered, so it cannot bind to the operator, so RNA polymerase can transcribe all of these structural genes. Now, when you're writing about this, I would recommend that you actually make a table and or do it as a flow chart. And the first question you ask yourself is lactose present? If it is, the repressor will be prevented from binding to the operator. If it isn't, the repressor will bind to the operator. There are a number of other ways that operons work. This is by far the simplest and most straightforward. I have taught the tryptophan operon before at HND level, so you may find yourself coming across the tryptophan operon. And also, as a hint for the joys of slightly off-colour humour at university, if you think that 3 letters are used as an abbreviation relating to the substrate involved in this operon. Lactose, it's the Lac Operon. Tryptophan, it's the Trip Operon. I will leave you with a fun thought exercise. What do you think we call the Foucault's operon? See you next time.